Hi, and welcome to Mitosis and Meiosis, presented by Ashton Bowerly, Sophie Short, and Ella Lennox. Let's get right into it and start off with Meiosis 1. The first step in Meiosis 1 is going to be interphase. Now, as we can see in interphase, we have our cellular membrane, and inside that cell membrane, we have our centrosomes, our nuclear envelope, and our chromatin. So chromatin is actually uncondensed DNA, which will later be used to form our chromosomes. So following interphase, we're going to see what we would call early prophase 1. Now in early prophase 1 is where we can really first see that there are two sets or pairs of homologous chromosome in this example. These pairs allow the cell to be what we would call a diploid cell, or 2N. Now in this example, because it is still early prophase, we can still see the nuclear envelope. However, it is fading fast and we will not be able to see it in the next examples. Another thing to notice is the spindle fibers are starting to form off the centrosomes, and these will be very important in the following phases of meiosis. In our next slide, we can see that the nuclear envelope has fully dissipated, and what is left are the two pairs of homologous chromosomes and the four pairs of sister chromatids. So each chromosome has two sister chromatids, and each chromosome has been replicated, so there are two homologous chromosomes. And the spindle fibers are still growing and will continue to lengthen as we go on and move forward in meiosis. Following prophase, we have what we call metaphase 1. In metaphase 1, spindle fibers are used to line up the chromosomes at the center of the cell, which we would call either the metaphase plate or the cellular equator. So I'd like to point out here that the spindle fibers use a process called independent assortment during metaphase. And this independent assortment means that as long as the homologous chromosomes are paired together and one is on each side of the metaphase plate, then that means it does not matter which individual chromosome is on which side. So this allows for more diversity in the gametes at the end of meiosis. Another process that has a similar job to this is what we would call crossing over. So crossing over is when homologous pairs share different alleles with each other to mix up or change the genes within the chromatids across from each other. So it does a very similar thing as independent assortment. It just diversifies the gametes at the end of the meiosis process. After metaphase, we have anaphase, where the spindle fibers break apart the homologous pairs and start to bring them towards their individual spindle poles at each side of the cell. Following this, we have what we call cytokinesis, where the cellular membrane pinches itself off, and as we can see in telophase 1, forms two individual cells. So I'd like to take a minute to identify what is still left in each of these cells. So as you can see, we have one centrosome each, which will soon be replicated and become two to further the process of meiosis 2. And we have two unidentical chromosomes in each cell. And each of those chromosomes have two unidentical sister chromatids. I'd like to point out that because we no longer have those homologous pairs, these cells are no longer diploid, they are haploid as of this stage of the process. So we are now done with meiosis 1, and we'll continue on to meiosis 2 in the next slide.
After meiosis 1, there are two haploid daughter cells that contain half the number of chromosomes than at the beginning of meiosis 1. The first phase of meiosis 2 is prophase 2, when the spindle apparatus begins to form. While this is happening, the chromosomes condense again and begin moving towards the center of the daughter cells. After prophase 2, metaphase 2 begins. In metaphase 2, the chromosomes line up at the metaphase plate. Unlike metaphase 1, in metaphase 2, each of the chromosomes is attached by the spindle fibers to both of the spindle poles. Then in anaphase 2, the sister chromatids separate by being pulled apart by the kinetochore microtubules and moving towards opposite poles of the spindle apparatus. In telophase 2, each sister chromatid moves to a different daughter cell and are now considered independent daughter chromosomes. After telophase 2, cytokinesis happens. During cytokinesis, the cytoplasm of the cell divides, although the number of chromosomes remains unchanged. Also during cytokinesis, the nuclear envelope reforms around the newly independent daughter chromosomes. The final product of meiosis II is 4 haploid cells that are also 1N.
Now we will look at the cellular process called mitosis. The goal of mitosis is different than that of meiosis. Instead, it will result in 2 sister cells that are the exact same. Just like meiosis, we will start this cellular process in interphase. Here, DNA is replicating and regular cell processes are occurring. Here you can see the cell membrane, chromatin, the nuclear envelope, and centrosomes represented on this beautiful cupcake. We will now enter mitosis, which is a process that consists of 4 steps. First, prophase. Second, metaphase. Third, anaphase. And finally, telophase, which is coupled with cytokinesis. During prophase of mitosis, we will see that the nuclear envelope of our diploid cell begins to fade away. As well, the chromatin will condense to form homologous chromosomes, which consist of 2 sister chromatids. The cell has begun metaphase when the homologous chromosomes line up down the middle of the cell. As seen, centrosomes will shift to opposite ends of the cell. From the centrosomes will reach spindle fibers. These fibers reach towards the metaphasic plate, then latching themselves onto a sister chromatid. Following metaphase is anaphase. During anaphase, the homologous chromosomes are split, resulting in 2 sister chromatids that will be pulled in opposite directions towards the centrosomes by the spindle fiber fragments. This is a big stage in mitosis because it will soon result in the formation of 2 separate cells. This leads us into the final stage of mitosis, which is telophase. In telophase, the nuclear envelope will reform around those sister chromatids. Once that nuclear envelope has been reformed, these sister chromatids will uncondense back into chromatin. For a period of time, this will result in a binucleate cell. As well, the spindle fibers will disappear due to the fact that they are no longer needed. Fairly often, cytokinesis is coupled with telophase. Cytokinesis is the splitting of the cell membrane, thus resulting in 2 sister cells. Now at this time, the sister cells are diploid. This quite incredible process results in 2 sister cells. Without this incredible process, we would quite literally be nothing. So thank you, mitosis. And thank you for watching this lecture on meiosis and mitosis, presented by group number 1. Have a great rest of your day.